Hi, in this video, we will discuss the generation of diversity of immune repertoire. And it will be divided into two parts, which is the B cell receptor, also known as antibodies, and T cell receptor. These two immune cells played different important roles in our body, where B cell receptor bound to molecules such as protein while T cell receptor bind to peptide MHC complex. Did you know, when we get sick, our body will fight against the bacteria or viruses by generating antibodies, and these antibodies are highly specific for different types of antigens. For instance, the antibody will only respond to one specific type of bacteria. Generally, the progenitor lymphoid cells are produced in bone marrow, and then they can differentiate into progenitor B cell or progenitor T cell. There are 5 main isotypes of antibodies in humans, namely IgG, IgA, IgM, IgE, IgD. Each of the antibodies would undergo somatic recombination to form specific types of antibodies, which lead to existence of estimately 10 billion types of antibody. Now, let's understand the structure of antibodies. Each antibody consists of 4 polypeptides, 2 light chains, and 2 heavy chains, joined to form a Y-shaped molecule. Each of the 4 chains has a variable, V, region at its amino terminus, which contributes to the antigen binding site, and a constant, C, region, which determines the isotype. The light chains are bound to the heavy chains by disulfide bonds. In heavy chain gene, it contains a leader segment, V, D, J segment, and the constant region. In the developing B cell, the first recombination event will occur between D and J segment. Any DNA between these two genes is deleted. This DJ recombination is followed by the joining of one V segment, forming a rearranged VDJ segment. All other segments between V and D segments are now deleted from the cell's genome. Primary transcript is generated, containing the VDJ region. The primary RNA is processed to add a polyadenylated, poly-A, tail after the C chain, and to remove sequence between the VDJ segment and this constant gene segment. After VDJ recombination of heavy chain, the light chain gene will undergo somatic recombination. The light chain gene is different from heavy chain gene, where the light chain gene does not contain D region, but it still contains V, J, and C segment. The first step of recombination for the light chains involves the joining of the V and J chains to give a VJ complex, before the addition of the constant chain gene during primary transcription. The RNA with V and J bound together will then undergo splicing, where the intron will be removed to form light chain protein. Finally, the products from VDJ recombination of heavy chain and VJ recombination from light chain forms the antibody structure. This is the summary of the number of genes for heavy chain and light chain genes, in order to generate the diversity of antibodies. T cell repertoire is the diversity of T cell receptors. In an environment with diverse pathogens, antigen-presenting cells continually sample various antigens. The unique nature of these antigens poses a challenge for TCRs, highlighting the necessity for a diverse T cell repertoire to effectively respond to the dynamic antigenic landscape. To create diversity in the T cell receptor, TCR, a process called gene rearrangement is at play. The alpha and beta chains of the TCR directly engage with antigens, and this interaction triggers VDJ recombination in the gene segments. T cells have different gene segments, alpha, beta, gamma, and delta, all equipped with VDJ components for recombination. This process requires signal sequences, RAG1 and RAG2 enzymes to kickstart the VDJ recombination. There are various V, D, and J segments. Let's say we have V segments from V1 to Vn, D segments from D1 to Dn, and J segments from J1 to Jn. In each recombination event, different combinations of D and J can come together to form a new type of TCR. Even though both cases have alpha and beta chains, these chains are a bit different from each other. This diversity in origin ensures that these two TCRs have distinct affinities toward antigens, which is crucial for an effective T cell response. VDJ recombination happens during T cell development in the thymus. The process begins with lineage commitment in the thymic cortical region. After committing to a lineage, T cells go through several stages, starting with the double negative stage. During the transition from double negative stage 1 to double negative stage 2, beta chain rearrangement occurs. After reaching double negative stage 4, alpha chain rearrangement takes place. This alpha and beta chain rearrangement occurs at different times within the thymus. Besides alpha-beta rearrangement, there's a possibility of gamma-delta rearrangement, although it's rare. This rarity is due to the need for 2 rearrangement events. Junctional diversity. Both B cells and T cells are undergoing junctional diversity and combination diversity. The difference is B cells involved heavy chain and light chain, while T cells involved alpha and beta chain. Rag complex binds to and cleaves recombination signal sequences to yield a DNA hairpin. Then, RAG-mediated cleavage of hairpin generates palindromic P nucleotides. N-nucleotide additions by terminal deoxynucleotidyl transferase. The complementary pairing occurs, unpaired nucleotides are removed by an exonuclease, the gaps are filled by DNA synthesis and ligation to form coding joint. Another is combination diversity. Additional diversity, termed combination diversity, results from the random recombination of separate V, D, and J gene segments to form a complete V region exon. For recombining genes of the variable region in alpha chain, there are 40 variable region gene fragments and 5 joint gene fragments. After restructuring, both fragments are joining to generate VJ combinations. 40 variable region gene fragments and 5 joint gene fragments can generate 200 possible VJ combinations for the TCR alpha chain. For recombining genes of the variable region in beta chain, there are 65 variable region gene fragments, 27 diverse gene fragments and 6 joint gene fragments. After restructuring, both fragments are joining to generate VDJ combinations. 65 variable region gene fragments, 27 diverse gene fragments and 6 joint gene fragments can generate 10,530 possible VDJ combinations for the TCR beta chain. There are 200 types of TCR alpha chain and 10530 TCR beta chain, which make as many as 210,600 different types. These are the summary of the difference of junctional and combination diversity between T cell and B cell.